What is biotechnology?
3, 2, 1, 0.
Biotechnology is the application of biological organisms, systems, or processes to develop products and technologies. Define genetic engineering.
3, 2, 1, 0.
Genetic engineering involves the manipulation of an organism DNA to alter its genetic makeup. What are transgenic organisms?
3, 2, 1, 0.
Transgenic organisms are those whose genomes have been altered by the introduction of foreign DNA. Name the first genetically modified organism.
3, 2, 1, 0.
The first genetically modified organism was a bacterium, Escherichia coli. Explain the concept of recombinant DNA technology.
3, 2, 1, 0.
Recombinant DNA technology involves the insertion of a gene or genes into an organism to produce new traits. What is a vector in genetic engineering?
3, 2, 1, 0.
A vector is a DNA molecule used to carry foreign genetic material into a cell. Name 2 commonly used vectors in genetic engineering.
3, 2, 1, 0.
Plasmids and bacteriophages are commonly used as vectors in genetic engineering. What is the role of restriction enzymes in genetic engineering?
3, 2, 1, 0.
Restriction enzymes are used to cut DNA molecules at specific locations, aiding in the insertion of foreign DNA. What is polymerase chain reaction, PCR?
3, 2, 1, 0.
PCR is a technique used to amplify a specific segment of DNA, making millions of copies in a short time. Define cloning in biotechnology.
3, 2, 1, 0.
Cloning is the process of creating genetically identical organisms or cells. What is a gene gun and how is it used in biotechnology?
3, 2, 1, 0.
A gene gun is a device used to transfer foreign DNA into plant cells by shooting microscopic particles coated with DNA. Explain the concept of gene therapy.
3, 2, 1, 0.
Gene therapy involves the introduction or alteration of genetic material within a patient's cells to treat disease. What are the applications of biotechnology in agriculture?
3, 2, 1, 0.
Biotechnology in agriculture includes genetic modification of crops for disease resistance, higher yield, and pest resistance. How has biotechnology been used in medicine?
3, 2, 1, 0.
Biotechnology in medicine includes the production of insulin, vaccines, and gene therapy. What is the role of bioreactors in biotechnology?
3, 2, 1, 0.
Bioreactors are vessels in which biological reactions take place, such as fermentation for the production of medicines or enzymes. Explain the significance of genetically modified crops.
3, 2, 1, 0.
Genetically modified crops are designed to have better nutritional content, disease resistance, and higher yield. What are biofertilizers?
3, 2, 1, 0.
Biofertilizers are living organisms that enhance the nutrient content of soil by fixing nitrogen and other beneficial processes. How do genetically modified plants help in pest control?
3, 2, 1, 0.
Genetically modified plants produce proteins that are toxic to pests, reducing the need for chemical pesticides. What is the significance of the Bt gene in biotechnology?
3, 2, 1, 0.
The Bt gene from Bacillus thuringiensis is used in crops to confer resistance to certain pests. What are monoclonal antibodies and how are they produced?
3, 2, 1, 0.
Monoclonal antibodies are identical antibodies produced by a single clone of cells, used in diagnostics and treatment. Describe the process of rDNA technology.
3, 2, 1, 0.
rDNA technology involves the isolation and manipulation of DNA to create recombinant DNA molecules. What are the applications of biotechnology in the field of forensic science?
3, 2, 1, 0.
Biotechnology is used in forensic science for DNA profiling, identification of individuals, and criminal investigations. Define the term bioinformatics.
3, 2, 1, 0.
Bioinformatics is the use of computational tools and techniques to store, analyze, and interpret biological data. Explain the role of enzymes in biotechnology.
3, 2, 1, 0.
Enzymes are used in biotechnology for cutting DNA, amplifying genes, and catalyzing various biochemical reactions. What is the principle of Southern blotting?
3, 2, 1, 0.
Southern blotting is a method used to detect specific DNA sequences by transferring them onto a membrane. What are restriction endonucleases?
3, 2, 1, 0.
Restriction endonucleases are enzymes that cut DNA molecules at specific sequences, producing restriction fragments. What is the purpose of ligases in recombinant DNA technology?
3, 2, 1, 0.
Ligases are enzymes used to join DNA fragments together by forming phosphodiester bonds between them. Name the first transgenic animal.
3, 2, 1, 0.
The first transgenic animal was a mouse, created by introducing foreign genes. What is the significance of the T plasmid in genetic engineering?
3, 2, 1, 0.
The T plasmid from Agrobacterium tumefaciens is used as a vector for transferring genes into plants. What are plasmids and how are they used in biotechnology?
3, 2, 1, 0.
Plasmids are small, circular DNA molecules used as vectors in genetic engineering to carry foreign DNA into a cell. Explain the role of the Agrobacterium tumefaciens in plant biotechnology.
3, 2, 1, 0.
Agrobacterium tumefaciens is used to transfer foreign DNA into plants. often for the development of transgenic plants. What is the role of the plasmid vector PBR322? PBR322 is a commonly used plasmid vector for cloning in genetic engineering, capable of carrying foreign genes. What is the function of the promoter region in a recombinant plasmid? The promoter region controls the expression of the foreign gene inserted into the plasmid. What are the benefits of Bt cotton?
3, 2, 1, 0.
Bt cotton is genetically modified to produce a toxin that kills certain pests, reducing the need for chemical pesticides. How is insulin produced through recombinant DNA technology?
3, 2, 1, 0.
Insulin is produced by inserting the human insulin gene into E. coli or yeast cells, which then produce insulin. Define bioremediation in the context of biotechnology. Bioremediation is the use of microorganisms or plants to clean up environmental pollutants. What are genetically modified (GM) organisms?
3, 2, 1, 0.
GM organisms are those that have been altered through genetic engineering to introduce new traits. What is a genomic library?
3, 2, 1, 0.
A genomic library is a collection of DNA fragments from an organism, stored in vectors, representing the entire genome. How is PCR used to amplify a gene of interest?
3, 2, 1, 0.
PCR uses cycles of heating and cooling to replicate a specific DNA segment many times, allowing for amplification. What are secondary metabolites, and how are they used in biotechnology?
3, 2, 1, 0.
Secondary metabolites are compounds produced by plants and microorganisms, often used for pharmaceuticals and agriculture. Explain the principle of gel electrophoresis.
3, 2, 1, 0.
Gel electrophoresis separates DNA fragments based on their size and charge by applying an electric field through a gel matrix. What is the role of the lac operon in gene expression?
3, 2, 1, 0.
The lac operon is a model for gene regulation in bacteria, controlling the breakdown of lactose when it's available. Define RNA interference, NEI, and its role in biotechnology.
3, 2, 1, 0.
NEI is a process where RNA molecules inhibit gene expression by neutralizing targeted mRNA molecules. What is the role of transformation in genetic engineering?
3, 2, 1, 0.
Transformation is the process of introducing foreign DNA into bacterial or plant cells to produce transgenic organisms. Explain the concept of transgenic plants.
3, 2, 1, 0.
Transgenic plants are genetically modified to include DNA from another species, improving traits like pest resistance or yield. What is Agrobacterium-mediated gene transfer?
3,
Agrobacterium-mediated gene transfer is a method used to transfer foreign DNA into plant cells. Using Agrobacterium tumefaciens. What is the significance of recombinant vaccines?
3, 2, 1, 0.
Recombinant vaccines are produced by inserting antigens from pathogens into a non-pathogenic organism, stimulating immunity without causing disease. How does recombinant DNA technology help in the production of human growth hormone?
3, 2, 1, 0.
Human growth hormone is produced by inserting the gene responsible for its synthesis into bacteria or yeast cells. What is an expression vector?
3, 2, 1, 0.
An expression vector is a plasmid designed to express a gene in a host cell, typically used for protein production. What is a synthetic gene, and how is it used in biotechnology?
3, 2, 1, 0.
A synthetic gene is a gene that is artificially constructed in the lab and can be used to produce specific proteins or enzymes. What is the significance of genetic fingerprinting?
3, 2, 1, 0.
Genetic fingerprinting is used for individual identification, parentage testing, and forensic analysis by analyzing DNA patterns. Define the term biotechnology in relation to medicine.
3, 2, 1, 0.
In medicine, biotechnology involves the use of biological organisms or processes to create therapeutic drugs and diagnostic tools. What is gene sequencing, and how is it useful in biotechnology?
3, 2, 1, 0.
Gene sequencing is determining the order of nucleotides in a DNA molecule, crucial for understanding genes and their functions. How does biotechnology assist in pest control?
3, 2, 1, 0.
Biotechnology helps in pest control through the development of pest-resistant crops and the production of bio-pesticides. What are the challenges in genetically modifying plants?
3, 2, 1, 0.
Challenges include gene stability, public acceptance, and unintended effects on non-target organisms. What is an insulin-producing transgenic organism?
3, 2, 1, 0.
An insulin-producing transgenic organism is one that carries the human insulin gene, enabling it to produce insulin. How is the E. coli bacterium used in biotechnology?
3, 2, 1, 0.
E. coli is commonly used as a host in biotechnology for cloning and protein production due to its rapid growth and simplicity. What is the difference between a plasmid and a virus as vectors in genetic engineering?
3, 2, 1, 0.
Plasmids are small, circular DNA molecules used for gene transfer, while viruses are natural carriers of genetic material and can also be used as vectors. What is the role of Bacillus thuringiensis in biotechnology?
3, 2, 1, 0.
Bacillus thuringiensis is used to produce a protein toxic to certain pests, making it useful in genetically modified crops. What is the principle of DNA sequencing?
3, 2, 1, 0.
DNA sequencing determines the nucleotide sequence of a DNA molecule using methods such as chain termination or next-generation sequencing. What are the advantages of using genetically modified microorganisms in the production of enzymes?
3, 2, 1, 0.
GM microorganisms can produce enzymes more efficiently and in larger quantities, reducing production costs. What is an example of a genetically modified plant with enhanced nutritional content?
3, 2, 1, 0.
Golden rice is a genetically modified plant with enhanced levels of vitamin A, aimed at combating vitamin A deficiency. Explain the term biotechnology in the environment.
3, 2, 1, 0.
Biotechnology in the environment refers to using living organisms, such as microbes or plants, to reduce pollution and enhance environmental sustainability. What are plant tissue cultures and how are they used in biotechnology?
3, 2, 1, 0.
Plant tissue culture involves growing plant cells or tissues in controlled conditions to produce plants with desired traits. What is the process of transfection?
3, 2, 1, 0.
Transfection is the process of introducing foreign DNA into eukaryotic cells, often used in mammalian cell culture. How do genetically modified organisms, GMOs, affect biodiversity?
3, 2, 1, 0.
GMOs may impact biodiversity by potentially crossbreeding with wild species or by introducing new traits that affect ecosystems. What is the role of cryoproteins in genetically modified crops?
3, 2, 1, 0.
Cryoproteins, derived from Bacillus thuringiensis, are toxic to certain pests and are used to protect crops from insect damage. What is the purpose of cDNA synthesis?
3, 2, 1, 0.
cDNA synthesis involves creating complementary DNA from mRNA to study gene expression and produce recombinant proteins. How does gene cloning work in biotechnology?
3, 2, 1, 0.
Gene cloning involves isolating a specific gene, inserting it into a vector, and multiplying the gene in a host organism. What is the role of ribozymes in biotechnology?
3, 2, 1, 0.
Ribozymes are RNA molecules that catalyze chemical reactions, and they are used in biotechnology for gene regulation and therapeutic applications. How is biotechnology used in the production of antibiotics?
3, 2, 1, 0.
Biotechnology is used to produce antibiotics by fermenting microorganisms that naturally produce these compounds, such as penicillium. What is the principle of Western blotting?
3, 2, 1, 0.
Western blotting is a technique used to detect specific proteins in a sample by separating them via gel electrophoresis and transferring them to a membrane. What is the role of hybridomas in biotechnology?
3, 2,
Hybridomas are created by fusing a specific antibody-producing B cell with a myeloma cell, resulting in cells that produce monoclonal antibodies. What are the ethical concerns surrounding genetic modification?
3, 2, 1, 0.
Ethical concerns include the potential for unintended environmental impacts, the safety of GMOs for consumption, and issues of patenting and ownership. Explain how genetic modification helps in biofarming.
3, 2, 1, 0.
Biofarming involves genetically modifying plants or animals to produce pharmaceutical products, such as vaccines or antibodies. What is the significance of the GFP, green fluorescent protein, in biotechnology?
3, 2, 1, 0.
GFP is used as a reporter gene to track gene expression and protein localization in living cells. How does biotechnology help in the treatment of diseases like cancer?
3, 2, 1, 0.
Biotechnology aids in cancer treatment through the production of monoclonal antibodies, gene therapy, and targeted drugs. What are the applications of biotechnology in waste management?
3, 2, 1, 0.
Biotechnology helps in waste management through the use of microorganisms to degrade pollutants and convert waste into useful products. What is the role of the hepatitis B vaccine produced by biotechnology?
3, 2, 1, 0.
The hepatitis B vaccine is produced by inserting the gene for the hepatitis B virus surface antigen into yeast cells to stimulate immunity. Explain the process of in vitro fertilization, IVF, in animals.
3, 2, 1, 0.
IVF involves fertilizing eggs outside the body and implanting the fertilized eggs into a surrogate mother, often used in animal breeding programs. What is the role of CRISPR-Cas9 in gene editing?
3, 2, 1, 0.
CRISPR-Cas9 is a powerful gene editing tool that enables precise changes to DNA, offering potential in genetic research and therapy. What are molecular markers, and how are they used in biotechnology?
3, 2, 1, 0.
Molecular markers are specific DNA sequences used for identifying genetic traits and are important in plant breeding and genetic mapping. What is the role of biotechnology in biofuels production?
3, 2, 1, 0.
Biotechnology helps produce biofuels by using microorganisms to convert organic materials, such as plant biomass, into energy sources. What are the benefits of using microbes in the production of enzymes?
3, 2, 1, 0.
Microbes can be genetically modified to produce enzymes more efficiently, enabling industrial-scale production of enzymes for various applications. How are transgenic animals used in the production of human proteins?
3, 2, 1, 0.
Transgenic animals are used to produce human proteins such as clotting factors, antibodies, and growth hormones for medical use. What is the role of biotechnology in food processing?
3, 2,
Biotechnology aids in food processing by using microorganisms for fermentation and preserving food, as well as enhancing flavor and nutritional content. What are monoclonal antibodies?
3, 2, 1, 0.
Monoclonal antibodies are identical antibodies produced by a single clone of cells, used for specific targeting in diagnostic and therapeutic applications. How does biotechnology contribute to sustainable agriculture?
3, 2, 1, 0.
Biotechnology contributes to sustainable agriculture by developing drought-resistant crops, reducing pesticide use, and improving soil fertility. What are bioinsecticides and how are they used in agriculture?
3, 2, 1, 0.
Bioinsecticides are insect-killing substances derived from natural organisms, such as Bacillus thuringiensis, used to protect crops from pests without harming the environment. Explain the process of creating a transgenic animal.
3, 2, 1, 0.
A transgenic animal is created by inserting foreign DNA into its genome, often through microinjection into a fertilized egg or somatic cell nuclear transfer. What is the significance of the Human Genome Project?
3, 2, 1, 0.
The Human Genome Project mapped the entire human genome, which helps in understanding genetic diseases, personalized medicine, and evolution. How are yeast cells used in biotechnology?
3, 2, 1, 0.
Yeast cells are used in biotechnology for the production of alcoholic beverages, bread, and biofuels, as well as for producing recombinant proteins. What is the difference between somatic cell nuclear transfer and embryo splitting?
3, 2, 1, 0.
Somatic cell nuclear transfer involves transferring a nucleus from a somatic cell into an egg cell, while embryo splitting involves dividing an early-stage embryo to create clones. How does gene therapy work in treating genetic disorders?
3, 2, 1, 0.
Gene therapy involves inserting a normal gene into the cells of a patient to replace a defective or missing gene, aiming to treat genetic disorders. What is the role of biocatalysts in industrial biotechnology?
3, 2, 1, 0.
Biocatalysts are enzymes or other biological molecules used to speed up chemical reactions in industrial processes, such as the production of biofuels or pharmaceuticals. How are GMOs regulated for environmental safety?
3, 2, 1, 0.
GMOs are regulated through environmental risk assessments, long-term monitoring, and safety testing to assess their impact on biodiversity and ecosystems. What are the applications of biotechnology in the textile industry?
3, 2, 1, 0.
Biotechnology in the textile industry includes the use of enzymes for fabric finishing, dyeing, and improving the quality of textiles without harmful chemicals. What is the principle behind the production of recombinant vaccines?
3, 2, 1, 0.
Recombinant vaccines are produced by inserting genes from pathogens into non-pathogenic organisms, which then produce antigens to stimulate immunity. How does biotechnology contribute to the development of biofertilizers? Biotechnology is used to produce biofertilizers by cultivating nitrogen-fixing bacteria and other microorganisms that enhance soil fertility. What is the process of DNA fingerprinting? DNA fingerprinting involves analyzing unique patterns in an individual's DNA to identify or distinguish between people, used in forensic science and paternity testing. What are the environmental impacts of using GMOs in agriculture? Environmental impacts include gene flow to non-GMO plants, the development of resistance in pests, and changes in biodiversity. What is the role of CRISPR-Cas9 in genetic modification? CRISPR-Cas9 is a gene-editing technology that allows for precise alterations in DNA, enabling genetic modifications in plants, animals, and humans. How does biotechnology help in the production of biodegradable plastics? Biotechnology aids in producing biodegradable plastics by using microorganisms or plant-based materials to create sustainable, eco-friendly polymers. What is the role of genetic modification in improving plant resistance to diseases? Genetic modification improves plant resistance to diseases by introducing genes that code for pathogen-resistant proteins or enzymes. What are the applications of biotechnology in biopharmaceuticals? Biotechnology is used to produce biopharmaceuticals, including insulin, vaccines, and monoclonal antibodies, through recombinant DNA technology. How does biotechnology contribute to environmental conservation? Biotechnology contributes to environmental conservation through bioremediation, waste treatment, and the development of renewable resources like biofuels. What is the role of biotechnological tools in agricultural biotechnology? Biotechnological tools such as molecular markers, gene sequencing, and genetic engineering help develop crops with desired traits like improved yield and disease resistance. How does biotechnology help in the production of therapeutic proteins? 3, 2, 1, 0. Biotechnology uses genetically modified organisms like bacteria and yeast to produce therapeutic proteins, such as insulin, clotting factors, and growth hormones. What are the ethical considerations in the use of biotechnology? 3, 2, 1, 0. Ethical considerations include the potential risks to human health, environmental impact, animal welfare, and the morality of genetic manipulation. What is a bioreactor and how is it used in biotechnology?
3, 2, 1, 0.
A bioreactor is a vessel used to cultivate microorganisms or cells for the production of biological products like enzymes, antibiotics, and vaccines. What is a gene library, and how is it created?
3, 2, 1, 0.
A gene library is a collection of DNA fragments from an organism, stored in vectors like plasmids or phages, used for gene identification and study. How is the polymerase chain reaction, PCR, used in biotechnology?
3, 2, 1, 0.
PCR amplifies small amounts of DNA, making it useful for gene cloning, diagnosing diseases, and forensic analysis. What is the role of biotechnology in improving livestock productivity?
3, 2, 1, 0.
Biotechnology improves livestock productivity through genetic modification for traits like disease, resistance, faster growth, and better reproductive performance. How is biotechnology used in the production of biofuels?
3, 2, 1, 0.
Biotechnology produces biofuels by fermenting plant material or using microorganisms to convert organic waste into ethanol or methane. What is the role of Agrobacterium tumefaciens in genetic engineering?
3, 2, 1, 0.
Agrobacterium tumefaciens is used as a natural vector to transfer foreign genes into plant cells by causing crown gall disease. What is bioremediation?
3, 2, 1, 0.
Bioremediation is the use of microorganisms or plants to degrade or detoxify environmental pollutants, such as oil spills or heavy metals. How are restriction enzymes used in biotechnology?
3, 2, 1, 0.
Restriction enzymes are proteins that cut DNA at specific sequences, allowing scientists to manipulate DNA for cloning and gene analysis. What is the role of biotechnology in the production of vaccines?
3, 2, 1, 0.
Biotechnology is used to produce vaccines by growing pathogens in culture, using recombinant DNA technology, or producing viral proteins in host cells. What is the difference between transgenic and genetically modified organisms?
3, 2, 1, 0.
Transgenic organisms have foreign genes from other species, while GMOs may include organisms with genes altered within their own species. How do genetically modified crops benefit agriculture?
3, 2, 1, 0.
GM crops can be engineered for higher yield, pest resistance, drought tolerance, and improved nutritional content. What is the process of gene silencing?
3, 2, 1, 0.
Gene silencing involves suppressing the expression of a specific gene using RNA interference or other techniques to study gene function. What are the applications of biotechnology in human health?
3, 2, 1, 0.
Biotechnology is used in human health for drug production, genetic testing, gene therapy, and vaccine development. How are DNA microarrays used in biotechnology?
3, 2, 1, 0.
DNA microarrays are used to measure gene expression levels across thousands of genes simultaneously. By detecting mRNA hybridization. What is the significance of stem cells in biotechnology?
3, 2, 1, 0.
Stem cells are undifferentiated cells with the potential to become different types of cells. Used in regenerative medicine and disease treatment. What is the function of the gene gun in genetic transformation?
3, 2, 1, 0.
The gene gun is used to introduce foreign DNA into plant cells by shooting microscopic gold or tungsten particles coated with DNA into cells. What is the role of biotechnology in environmental cleanup?
3, 2, 1, 0.
Biotechnology helps in environmental cleanup through the use of microorganisms or enzymes to break down pollutants. A process called bioremediation. What is the significance of cryogens in biotechnology?
3, 2, 1, 0.
Cryogens, from Bacillus thuringiensis, produce proteins that are toxic to certain insects, making them valuable in the development of pest-resistant crops. What are genetically modified mosquitoes used for?
3, 2, 1, 0.
Genetically modified mosquitoes are engineered to reduce the transmission of diseases like malaria and dengue by either sterilizing them or making them resistant to pathogens. How does the process of gene cloning work in microorganisms?
3, 2, 1, 0.
Gene cloning in microorganisms involves inserting foreign DNA into a plasmid vector, then introducing the plasmid into bacteria that replicate the gene. What are the advantages of using genetically modified crops in developing countries?
3, 2, 1, 0.
GM crops can provide higher yield, better resistance to pests, and improved nutritional quality, which can help ensure food security in developing countries. What is the role of biotechnology in improving livestock?
3, 2, 1, 0.
Biotechnology improves livestock by enhancing disease resistance, growth rates, and milk or meat production through genetic modification. How is recombinant DNA technology used in the production of human insulin?
3, 2, 1, 0.
Recombinant DNA technology involves inserting the human insulin gene into E. coli or yeast cells, which then produce insulin for diabetes treatment. What is a transgenic bacterium?
3, 2, 1, 0.
A transgenic bacterium is a bacterium that has been genetically modified to carry foreign genes, enabling it to produce proteins or other products of interest. How does biotechnology help in the conservation of endangered species?
3, 2, 1, 0.
Biotechnology aids in conservation by creating gene banks, using cloning techniques, and preserving genetic diversity in endangered species. What is the role of biotechnology in producing biofuels? Biotechnology helps produce biofuels by using microorganisms to ferment plant sugars into ethanol, or by using algae to produce biodiesel. What is the use of biotechnology in forensic science?
3, 2, 1, 0.
Biotechnology is used in forensic science for DNA profiling, which helps identify individuals in criminal investigations and paternity testing. What is the process of somatic cell nuclear transfer?
3, 2, 1, 0.
Somatic cell nuclear transfer involves transferring the nucleus of a somatic cell into a nucleated egg cell to create a clone of the donor organism. How does recombinant DNA technology contribute to the development of vaccines?
3, 2, 1, 0.
Recombinant DNA technology is used to produce subunit vaccines by inserting genes encoding pathogen proteins into host cells, which then produce these proteins for immunization. What is the role of biotechnology in improving crop yield?
3, 2, 1, 0.
Biotechnology improves crop yield by developing genetically modified plants that are resistant to pests, diseases, and environmental stresses, resulting in higher productivity. What are molecular diagnostics, and how are they used in biotechnology?
3, 2, 1, 0.
Molecular diagnostics use techniques like PCR and DNA sequencing to detect genetic disorders, pathogens, or mutations at the molecular level. How does biotechnology contribute to the production of antibodies?
3, 2, 1, 0.
Biotechnology enables the production of monoclonal antibodies by fusing specific immune cells with cancer cells to create hybridoma that produce large quantities of antibodies. How does the use of biotechnology in agriculture reduce the need for chemical pesticides?
3, 2, 1, 0.
Biotechnology allows for the development of pest-resistant crops, reducing the need for chemical pesticides and promoting sustainable farming practices. What is gene therapy, and how is it used to treat diseases?
3, 2, 1, 0.
Gene therapy involves replacing defective genes with healthy ones to treat genetic disorders by directly altering the patient's genetic material.